We will continue our discussion about deterministic barcoding in tissue, and this is the part 3 in which we will analyze the results from the DBiT from the already exist technique that measures spatial transcriptomes. So first what they have done in the panel A is that they need to check the length of the base pair that are predominantly found from this DBiT method, and from this from these results they come to a point that it is 100 base pair would be the optimum read length that can be used in the NGS sequencing, as the 100 base pairs close to 100 base pairs read length are more predominantly found. And thereafter what they did is they compare the number of genes that are produced, the number of genes that are found in different samples in different methods, and compare it with the DBiT-seq. So in the spatial transcriptomes, number of genes are almost similar. However, the difference is that the spatial transcriptomes are done on 100 micrometers, whereas this DBiT-seq and slide-seq are done on 10 micrometers. So these are not 100, 50 micrometers. So in the 10 micrometers, the number of genes that are found are almost comparable to the spatial transcriptomes, whereas for slide-seq the number of genes are lower compared to this DBiT-seq or the spatial transcriptomes. And when they check the unique molecular identifier, they can see that it is also comparables, or we can say the unique molecular identifiers are higher than this slide-seq. However, the difference is that the spatial transcriptomes are done on 50 micrometers, whereas DBiT-seq and slide-seq are done on 10 micrometers microfluidic width. And in the paper they have mentioned that this spatial transcriptomes genes are slightly higher than the DBiT-seq, but this can be compensated by the microfluidic length that have used in the spatial transcriptomes compared to DBiT-seq. In the spatial transcriptome they use 50 micrometer width, whereas in the spatial transcriptomes, whereas in the DBiT-seq they have used an micrometer width microfluidic chip. And this is they talked about the number of genes and unique molecular identifiers, and they talk about this how the this number of genes that are detected in different techniques compared to the number of reads that are being identified. So in this case they find out that the 50 micrometer microchannel width and the 10 micrometer width are almost saturated similar at the same time. However, they are higher than this spatial transcriptome techniques. And in this inner figure is also about the unique molecular identifier by read length, and unique molecular identifiers are higher in the case of this is what the paper say: unique molecular identifiers are higher in the case of the spatial transcriptomes compared to the this DBiT-seq with 10 micrometer channel or 50 micrometer channels. And within this DBiT-seq also, the different, when we change the microfluidic width, the number of unique molecular identifier increases. That can be seen here, and this can be because the pixel size increases, and now they can get more area to cover, and detect these gene and unique molecular identifiers. Next, they did a cumulative analysis. In the cumulative analysis, they find out that the percentage UMI, around 20 or top 20% of the genes covered just the 70% of the UMI, the unique molecular identifier, and whereas the top 10% of the genes covers 70% of the UMI. The top 30% of the genes covers 90% of the UMI, and this is same for almost all the techniques that are being used. And in the panel they shows that the biasness of this DBiT-seq and the other techniques such as spatial transcriptomes, that they are biased towards high length gene compared to the low size gene. And this is true not only for DBiT but also for spatial transcriptomics. So after analyzing, comparing the results with other techniques, they also need to check the results. They also need to check the results for this. They compared the results with immunostaining. They compared the result with immunostaining and their uh, DBiT labeling. And first they used to stain. First they stain this TTN using immunostaining, and thereafter they analyze this TTN from this DBiT, and they can find out that the TTNs are almost similarly overlap with this immunostaining, and they when they quantify the results, they are almost similar. The difference could be because of the different tissue slides that they have used, maybe the adjacent tissue slides. And same thing they have observed for SRP2 immunostaining, and this can be seen here, and for other compounds also, such as TRF immunostaining, uh, DLK1 immunostaining, that can be seen here, that all of them are almost matching. The minor difference could be because of the different tissue slides that they have used. And finally they compared the transcript count in the each pixels, and they can see that the transcript counts are slightly lower compared to this missed fish. And they the paper provided an explanation that this could be because of the same reason, that is different tissue slides that are used. So I hope you got an idea about this DBiT performance, and what are the characteristics, or what are the things that needs to be considered while preparing the samples for DBiT for NGS sequencing. If you need to understand more about this DBiT uh, techniques, or this DBiT spatial transcriptomes method, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.